What's up in the lab?
Genes are the coded messages that tell cells how to behave. Mutations in certain genes can cause them to divide uncontrollably, turning healthy cells into cancerous ones. And new ways of treating cancer can never come soon enough. Set up in Moscow in 1951, the Nikolai Blakhin Cancer Research Center is the largest such medical institution in Europe and second in the world. The center employs around 4,500 people and has over 1,200 beds for patients. It also conducts leading-edge experimental research in diagnosing, treating, and preventing all types of malignant tumors and systematic diseases, of which there are over 200.
There are only a few treatment methods. You can count them on one hand. Firstly, there's surgical removal of the tumor. Secondly, there's radiotherapy, that is, without removing the tumor, or radiotherapy after the removal of the tumor. And thirdly, there's systemic treatment, or chemotherapy.
In addition to the staple 3 approaches, different techniques are also being tried out. Biological therapies use vaccines or bacteria to encourage the body's immune system to fight cancer cells and reduce the side effects of more aggressive treatments. Tests have also been conducted using nanofiber tubes to draw tumors away from inoperable areas, potentially opening up new treatment opportunities. And the first concrete evidence of cancer stem cells might provide a way of destroying cancer at the root.
Molecular biology has given us an understanding of the specific genes and their derivative proteins that play a vital role in the formation of tumor cells, actually making a cell malignant and therefore different from a regular cell. Once we know these genes and understand how their proteins work, we can start developing targeted medicines for them.
Bind Rus was set up between Bind Therapeutics from Massachusetts and Rus Nano in 2011, with the Russian side investing $47.25 million. Bind Rus recently opened an R&D center at the Slava Technopark in Moscow. The focus of the company's activities will be perfecting its new targeted cancer treatment.
Our company's scientists are working on several in-house projects aimed at developing the next generation of the innovative drug group we call Accurins. These are targeted, programmable cancer drugs for the treatment of various types of cancer.
Accurins are like 100 nanometer Trojan horses. They consist of a therapeutic payload in a protective biocompatible polymer shell, which both controls the release of the medicine and protects it from the body's immune system. Once injected, the Accurins course through the bloodstream until they reach their target. Their size means that they can exploit the gap between the cells in the tumor's blood vessels, which is larger when compared to healthy vessels. Special ligand molecules covering the surface of the Accurin bind to the cancer cells and penetrate through the cell wall. Once inside, the toxic payload is released, destroying the cell from within. Producing the Accurins is relatively simple. The polyethylene glycol used to form the outer shell is dissolved in the medication. Accurins can be used to transport various medical payloads, but the company's most promising Accurin is Bind 14, which uses the tried and tested anti-tumor drug docetaxel. The polymer layer automatically self-assembles into nano shells around the drug. But what makes Bind 14 really stand out from other nanomedications are the ligands on the surface. These molecules actively target the proteins found on certain cancer cells and new blood vessels in tumors. This combination of active and passive targeting means that the toxicity of medication can be increased while reducing the risk to surrounding healthy tissues, potentially lessening some of the serious side effects associated with other types of chemotherapy. Once the particles have been produced, the 100 nanometer Accurins are separated from the rest of the mixture. This is the optimum size for distribution within the body and penetration of the tumor, but effectiveness isn't the only consideration.
The technology behind any new drug needs to be scalable. You must be able to produce grams or even kilograms of the drug just as well as you can produce it in milligrams. What makes the bind technology so efficient is the fact that it is so easily scalable.
If the occurrents are too small, they would be filtered out by the kidney. Too large, and they may not breach the tumour. So the solution is scanned to ensure they're within the 100 nanometer range and also to check the rate the medication is released. Preclinical studies have shown that Bind-14 significantly increases the concentration of docetaxel in tumours, boosting anti-tumour activity with no increase in toxicity. Bind-14 is now undergoing stage 2 clinical trials in the US and Russia. This will determine the optimum dosage and assess its effectiveness against specific types of cancer. Targeted therapies represent the future of cancer treatment. The high effectiveness of Bind-14, along with its ability to be mass-produced, means that we may soon have another weapon in the fight against cancer.